Hi everyone, welcome to our 11th session of the MedAI group exchange sessions. This week we actually have Florian Dubost here with us to talk about his research on data augmentation for regression networks. Florian is actually a postdoc in the Rubin lab here at Stanford and is affiliated with the Department of Biomedical Data Science. He holds a PhD in medical computer vision and has worked on numerous deep learning projects involving MRI, fMRI, and recently EEG signals and video data as well. So thanks so much, Florian, for joining us today. Before we start, do you have any preferences on how you would like us to ask questions? Like, is interrupting you fine, or would you like it at the end?
Thank you for the introduction. Yeah, you can interrupt me when I'm presenting, no problem, or you can keep it at the end, like you can do as you want.
Awesome. Okay, so let's try to make this session as interactive as possible. And without further ado, let me give the stage to Florian.
Thank you. So everyone sees the slides?
Yes.
Yeah, okay. So today I'm going to present a work that I did with my colleagues in Rotterdam before starting the postdoc. And it is entitled Hydranet data-augmentation for regression neural networks. And that's something we published in MedAI in 2019. So if you want, after the presentation to see more details, you can always check out the paper. It's on arXiv also. So the motivation of this work is to develop a method for small data sets where there are only image level labels. So you don't have segmentation, you don't have pixel-wise information. So it's for images, you don't have pixel-wise informations. You just have whatever training samples and a label for that training sample, but not like fine-grained. And these labels, they can be noisy, and that's actually I'll come back to this later. But if you think, for example, in medical data sets, you could have, if you want to estimate the volume of an organ or of a lesion, there will be some variation from one method to another, or from one rater to another. So the ground truth labels, they can be noisy, like by definition almost. And so if you have all of these factors, small data set, only image level labels, and the labels are noisy, you can very easily overfit to your training set and have a poor generalization performance. And that's what we're going to propose a method in this work to prevent that. So a form of regularization. The method that we are going to propose, it revolves around the combination of samples, of training samples. So there are two main motivations behind this, is that you can create more samples by recombining existing samples, like virtually you create more samples. So you virtually increase the size of your training set. But most importantly, you can average the noise in the label. So because now it's a bit when you do, like mini batch gradient descent, you average the noise, you average the error across samples. So instead of being concentrated on single sample, it can be averaged over the number of samples you use to do the combination. And there is one work that does this already, and it's called mix-up. And the way it's done is that so I put some picture from some figure from the article here, with some pseudocode and with some little figure. I think what's most important here is that or they construct these samples. They basically have this lambda parameter, and the X is the training sample, the Y is the label, and they basically just do a linear combination of two different training samples and their corresponding labels to create new samples. Okay. And that's the whole idea of the method, and it shows that it's a good realization technique. It improves generalization error. And you can see the figure on the right, like it sort of smooths the decision function. So we're going to do something that's a bit similar. One of the main differences is that we have another hypothesis that is that our labels, they should indicate a countable quantity. So that if you take two training samples and you sum them up, their labels should also be summed. That's our, like, there's no such hypothesis for mix-up. For like the intuition of our work, that's what we assume, so that you can basically combine training sample and sum them, and you will expect their labels, the corresponding label of the batch, to be also summed. And like, it would be meaningful for the task. And so the key concept is going to be that. So if I take some like real sample on the left, for example, it could be, you know, an MR image with different lesions, but it could just be like this image with dots, and you want to count the number of dots. Then if you take the first image that has 3 dots and the second image that has 2 dots, so this image would be the training sample X, and this would be the label Y. Then you can recombine them, and it, you know, if you look in the collection of images, it makes sense, you know, like you can count there are 5 dots. And this is your new virtual sample. And you can do that with all the combination of images. And if you want the original images, you know, you can just add an image that is where there is no dot, black image, and that you would still end up with the original sample. And you're not limited to just 2 samples, right? You could just sum all of them and have an image that has 11 dots, right? So if we formalize it a bit more, so each of these images, instead of X, I call it I1, I2, IN. So we select n images. So in the example before, n was equal to 2, but as I said, you could have n as large as you want, you know, versus like, whereas mix up, it was just 2. So in our approach, you can just take it as high as you want. And then you compute the label as the sum of the sample. So there's no, it's not a linear combination of lambda as in mix-up, it's just the sum of the labels. And you can think as the number of samples that you draw to do this combination as the regularization strength that you introduce. So if you have n equal 1, it would be like you don't actually use that method, right? It's just the baseline method. You compute the loss, like for the label is just the label of the sample. And the higher n will be, the more you're going to average training samples, basically. So if you do this method, you can create this amount of combinations. So you can trust me on this. And so this is just a suggestion, but usually we'll be dealing with a loss function that is mean squared error or mean absolute error between the label of the images or batch of images and the real and the predicted label, the label predicted by the network. So now there's the very first idea that comes to mind is that okay, what's the difference actually with mini batch stochastic gradient descent? Because that's the same, you know, you compute in a batch, you compute the loss across several samples, and then you use it to compute the updates. You use the average to compute the updates. Well, the difference is that so on the left, that would be mini batch stochastic gradient descent. So you do the sum after you computed the loss. In our case, it's the opposite. We first do the sum of the label and then compute the loss. And if your loss function is nonlinear, for example, mean squared error, this is not equivalent. So now the implementation. There are two ways to implement it. You could just modify or you compute, you know, the loss on during the training procedure, or, and that's what we did, it's maybe a bit more complex, but it's a bit easier to understand visually at least. You can just have this, what we call base regressor network on the left, where you have the input is the image, the output is this label that represents a quantity. And then a series, you know, of convolution, max pooling, subscheme connection, global average pooling, and fully connected layer. So this is just our base model. That's what we would use. And then we can just replicate these models as many times as we have input in our collection of images. So if you remember earlier, I called it n. So this would be here in that case, n equals 4. And then the weights between all of these base regressors, they are shared. So it's exactly the same network that's replicated 4 times, okay? And then you just have a summation layer at the end to aggregate the labels. And that's the output of your network. So to keep the original label in images, we're going to do what I mentioned earlier, adding black images. So let's say we formalize that our big network is called G. It takes S, the batch of images, so the batch of n images, I1, I2, IN. And because the network, they are all the same, you can write it like that. The sum of Fi, where F is our base regressor, Fi of small I. Sorry, no. So if all the Fi are different, that you can write it like that. But considering that all the F are the same, you can just have just one F that is our base regressor, and that's going to be the sum of the labels. So that's what we defined earlier. Okay. So if you look at line 2, now we could say, okay, all the images are black images with F of B. So B is a black image. F of B equals 0. Then you're just left with this image. So you can have, you know, the G of S, that's just F equal to F of IJ. So this is just equivalent if you add the regressor with one image, the baseline. If all the black images are, if you give all but one black image. And in our approach, we actually have a probability to draw a black image for each of these I1, I2. So the situation where they are all black can occur. And that's also, by the way, a parameter that you can use to influence the regularization. So because it's going to modify the value of the average value of n, basically. So I'm going to show some results, some experiments and results on 2 different tasks that are related to analysis of brain MRI images. The first one will be quantification of enlarged perivascular spaces, and the second one is the quantification of white matter appendices. And they are both different types of brain lesions that you can see on MRI. The first one, enlarged perivascular spaces. There are some little vessels in your brain, like you can see on the left image here. And there is a space between the vessel itself and the envelope of the brain, and that's the perivascular space. And sometimes this space, it can enlarge, and then it becomes visible on MR image. For example, on the right, this is this little hyper-intense structures. And so when it's enlarged, you can see it, and then you can quantify it. And here in the middle, there is reconstruction of the hemisphere of the brain, and you can see the little segmentation of these perivascular spaces. So you see it as a sort of tubular structure because it follows the blood vessels that are basically some sort of cylinders, right? And so the motivation to quantify these enlarged perivascular spaces that they are related to several small vessel diseases, such as stroke and vascular dementia. So the first task, it is to count the number of enlarged perivascular spaces in the basal ganglia. Basal ganglia, this is brain region that's more or less in the center of your brain. And we use the data set that's the Rotterdam scan study. So we actually use a subset of that because this is a very large data set. And this is some images of the basal ganglia that has been segmented in actual view. And you can see this little white hyperintense structure that are circled in red. That's what we want to quantify. So on the left, it's an image where there are few. On the right, it's an image where there are a lot of them. And you can see that it's not that easy, right? I mean, these were circled and were identified as perivascular spaces, but there are a lot of other little hyperintense structures. And it's not, when people quantify perivascular spaces, they basically choose a threshold, like a visual threshold based on the image intensity. So if it's very white, then they will for sure count it as perivascular space. If it's more gray, they won't. On T2 MRI, these images are T2 MRI. And yeah, it's not easy to choose that threshold. So it's not only about the threshold. People actually also need to scroll up and down to see the 3D structures. I mentioned earlier, it needs to be tubular. So it's not an easy problem, and there can be a lot of intra-rater variability in this kind of quantification. So for this task, we don't want to detect it or segment it. We just want to count the number of perivascular spaces. So on the left, that would be like, for example, an input to the network, and the label, it would be 1, 2, 3, 4. So exactly like the little image I showed earlier of the dots, this would be 4 dots. And on the image of the right, it's more dots. I'm not going to count it, but maybe like 20 or something. Yeah. So if we do that, so here you can see a plot that summarizes the results for different training with different amount of training samples. And on the y-axis, this is the mean square error between the automated and visual scores. So automated scores is the output of the network. Visual scores, it is the count that was made by human experts. And so you will see, like for all methods, the trend that, of course, the mean square. So the mean square error, the lower it is, the better, right? It's an error. So the smaller the error, the better it is. So you see that the error reduces with the number of samples. That's what you could expect. And we have 2 baselines. So in blue, this is our methods. And in red and green, this is 2 baselines that only use the base regressor F, so one of these, branches. And the blue one is yeah. There was a question. Okay. So the red one, it is exactly the same, and the green is exactly the same base regressor with adding a dropout at 0.3. I think after every convolution.
I have a question here.
Yeah.
So the x-axis is number of samples. Is it like number of fMRIs you use?
Yeah, so it's not fMRI, it's like structural MRI. So
Oh yeah, I mean, yeah, that's SMRIs. And what is the like the fake samples per batch here?
Yeah, so good. Yeah, I didn't mention it. This in that case we use 4 like batches of 4 images. So
Okay.
you would have to compute the number using the formula I gave earlier. Like you would have to use this.
Oh right. Okay, I see. And so from this figure, it seems like adding dropout to the base regression actually. Oh, okay. Sorry, this is the MSE loss. I see.
Yeah, so in that case, actually, it doesn't. So this
Oh,
is
it
the
doesn't
MS.
help. Yeah.
Yeah, so that's a good observation. Yeah, in that case, the dropout doesn't help the network. It actually makes it worse. So this is MSE on the test set, right? So not validation or training on a separate test set. So yeah, it doesn't help in that case, which is interesting because you would expect that dropout, as this regularization effect. That's why we did it in that case. We thought, okay, it's going to be better than the red line, but no, that didn't help. Maybe it could have been better if we decreased the dropout rate, to 10%. Maybe that was too high. I'm not sure. But yeah.
Okay, thanks.
You're welcome.
Can I ask one more question?
Yeah.
If you could go back to the previous slide.
This one?
So yeah, do you do any kind of pre-processing to make sure that the brain matter actually coincides between the samples when you add, or does it not matter? Like the background.
Yeah.
In your example, for instance, you had only the dots in a black background, so you can imagine like it's going to be a cleaner version of the mix-up. But now that you have the brain scan present, like would that lessen the value of, or lessen the contrast of the white dots?
Yeah. So the pre-processing on that, they're not registered, if that's the question. Like they're not elastically registered. Yeah, and with that, they are not even at all registered actually. So it's just I do the segmentation, okay. The basal ganglia, it has a different shape for different people, as you can see on this example. And then I just crop. So if you would take the brain MRI image, it would be larger than that. But if you just segment the basal ganglia, you would end up having a lot of black pixel around it that have no information. So what I do is that I compute the center of mass of the mask, and then I crop around this center of mass with a fixed size. So that's the processing. So there's nothing that forces it to be in the same place, and also it's intensity rescaling between 0 and 1 using the minimum maximum values. But that's about it. But then I do some data augmentation. So for all methods, all experiments, there is some data augmentation, such as translation, rotations, flipping. So that also helps for both the proposed method and the baseline. Yeah. So there is nothing that's that forces the different basal ganglia to have the same outline, like from one example to another. But I think it doesn't matter very much because you have this data augmentation. The algorithm is able to still pick up just the hyperintense region. So it's more difficult indeed than the case I showed with the little dots. But that's not a problem. Yeah.
Okay, thank you.
You're welcome. Yeah, so that's basically the results. So I'm going to now discuss the white matter hyperintensity. So white matter hyperintensities, they are white, so that's why hyperintensity areas in the white matter. So white matter refers to the actual brain structure, and hyperintensity is because it's white areas. So here you have some, and they are related to, again, cerebral small vessel disease, stroke, vascular dementia, same project line. So you see, below there are some example of different brain images, different load of, actually it's different slices. So, you know, even within one person, you could have such variety. But at least, you know, the left slice, there is not that many white matter hyperintensities. So it's delineated in yellow, but what you want to see is really this white area. So this is on FLAIR. On FLAIR, earlier I showed images, there are T2 images. This is FLAIR. If you see hyperintense region on FLAIR, it is like by definition, the white matter, it is by definition white matter hyperintensities. So there is not, but you still have to, you know, choose a threshold because sometimes it's not going to be, you know, very easy. In this case, it's not. It's pretty obvious. But like for example, this little area here, it's a bit hyper intense. Okay, do you segment it or not? You know, because like this, they were segmented. So it's also, there is also some variability in the label that depends on the rater, which creates noise in the labels. Like the same image, they won't have the same label from one rater to another. Yeah. So here you see a larger load, and here even larger. So for this experiment, we wanted to estimate the volume. Well, earlier, we just wanted to count the number of lesions. Here, we want to estimate the volume because it's not that informative necessarily the count of white matter hyperintensities, because there can be some which are very small. If I go back to the earlier slide, you see this is very small, so you count it as one, but this is also like much bigger, where you also count it the same when you want to quantify the white matter hyperintensity load. No, it wouldn't necessarily be, I mean, you could for some of the metric, but you would definitely not capture all the information. Whereas perivascular spaces, their size is it doesn't vary as much. It's pretty similar from one perivascular space to another. So that's why here we're interested in the white matter hyperintensity volume. So for this experiment, we used a public data set from the white matter hyperintensity challenge in MICAI 2017, which has 60 scans. You can see the website here. And there's a paper that summarizes the challenge results. So this is a summary of the results. So the method, the base method, so the base regressor F, which was the like this red line here, and blue, this is the proposed method. So in the first experiment, so experimented with different number of training scan versus testing scan. So training includes training and validation. Testing time are completely left independently. So, and it's each kind for a different subject. Same in the experiments with enlarged perivascular spaces. So we experiment with varying size of training set and testing set. So yeah, we couldn't use 30 anymore because there are only 60 images in the set. So in this case, it's a bit less accurate, the evaluation of the generalization error. And we also varied between the mean squared error loss function during optimization and the mean absolute error during optimization. So for the standard scenario, we have on average better performance with the method, and a lower variance. Whereas for the baseline method, it was worse on average, but the variance was much larger. So it could happen that by chance, depending on your organization or you show the sample to the network, it could have like similar results than the proposed method, but it's not. It depends on your parameter settings. Also be much worse in that case. So that's better to have something that has less variance. In that case, so we didn't have time to replicate the experiment in that case. So we just had one training, and it was much larger. And when we increased the number of training scans, it actually reached like the baseline method was a bit higher. We didn't compute whether this was significant or not, but you see that it starts to get similar results. Yeah, it was with the mean absolute error. Okay, so that's the last slide with some discussion and the conclusion. So I presented, this approach where I replicate the base regressor to compute this ginormous network. Like another way to implement it, as I mentioned, would be to change the computation of the loss over the mini batch. So you would just have the output. So you would not compute the loss directly. You just have the output of each of your samples in the mini batch, then sum them, and then compute the loss over that. And then you don't need to change the architecture of your model, right? You can just do that. So also eventually you have to remember that the number of images that you can process at the same time, it will be limited by your GPU memory. So if you have a big network like large 3D images, you won't be able to use a high N, like a lot of images in a batch to aggregate their labels. That's one of the reasons why in our experiments we didn't use more than 4 images in a batch, and why also our networks are pretty small. So the main, I think, idea behind this work is that combining label, it averages the label noise. The intuition originally I used to develop this method was that it creates virtually more samples. So you could think that, more samples, better generalization error. But I think the added value is that it averages the label noise more than just created virtual samples. And like an interesting fact is that for the quantification of perivascular spaces, we also computed inter-rater agreement of these visual counts. And we could show that just with 30 scans of this method and just one label per scan, so 30 scans, 30 labels, we are able, within a few hours of network training, we are able to reach the expert performance in terms of intraclass correlation coefficient. Yeah. So that's it. Thank you very much for your attention. Maybe it's a bit shorter than what was planned, but there's then more time for questions.
Thanks, Florian. That was really good. Maybe I can start off the questions with one of my own. So when you presented the white matter, like where you actually combine the volumes instead of the counts, were there cases where the intersection could mess with the combination? Like, for instance, if you're combining the second image and the third image, like many of the volumes could actually be within the second one's volumes. Does that make sense? So the sum will actually be less, like should be less than the or the combination should actually be less than the sum of either, but your label will be just the actual sum of the two. Does that make sense?
So I'm not sure I get it. So you saying that like these two images, they have very like this in this image there is very few and this image very many.
No, so I'm saying, so when you're actually combining the, let's say the second image and the third image, so you are also just adding a linear combination of the volumes to get the volume of the final mix-up, right?
Yeah, it's actually not like it's even really a sum, not even, I mean, you can consider a sum being a special case of linear combination, but yeah, we don't use coefficients. It's the coefficients are one.
So let's say you have the second image and the third image, and you're combining them to get a fourth image. The fourth image's like volume is actually going to be less than the actual sum of the second image volume and the third image volume, because some of the volumes are intersecting. Or am I reading this wrong?
No, so that's okay. So you're like this would be in the case of mix-up where they really like sum each voxel to like from one image to another, and in that case, yeah, it would be right, and that's why I don't. That's why the mix-up intuition is a bit strange, you know. When mix-up works in practice, but I think people don't understand exactly why yet. So what we do is different. We never sum the images. So if I go back to mix-up, so yeah, mix-up it does sum the images, right? But like so the voxel image intensities. But in our example, we don't sum them. We just like aggregate them. We sum the labels only. So if I go here, each image is going to be fed to a different head of the network. So in that case, if you have two images, there will just be two branches, and they will be processed independently.
I see. Got it. Okay.
Yeah.
Makes sense. Thanks.
So we don't have this problem of yeah, I mean your argument makes complete sense for mix-up. That it's difficult to interpret what it means, but for our method, that's not the case.
So that's actually a plus point for your method compared to mix-up.
Yeah, it's more interpretable. Yeah.
Got it. Thanks.
It's more interpretable, but it's the negative point is that it's as I mentioned, you know, earlier. It's restricted to labels that indicate a countable countable quantity. So if you have, I don't know, for example, if you're doing, if you just do classification of the tumor types from MR images, it won't necessarily make sense to use our method, because what does it mean, you know, adding several images? Whereas mix-up, I think in that case it could help. But yeah, then the intuition is not clear why it helps. So mix-up works in more cases, but people don't really know why. Our proposed method works in, I mean, I didn't test it in other cases, to be honest. So I know that it works in fewer cases, but it's clear why.
Got it. Okay, thanks. Does anybody else have other questions for Florian? Okay, so looks like we don't have more questions. But
actually, I have a question about like experimental detail. So your method is basically increasing the batch size or the sample size per batch. So in your comparison to the baseline, do you actually make sure that the batch size is the same?
Yeah, that's a good point. Yeah, I do. So it's batch size of 4 for these two, and a batch size of 4 for the blue method also. But instead of they go all at the same time through the network instead of consecutively. I mean, the GPU, it goes consecutively anyways. But yeah, so the batch size is the same. Yeah, that's a good point. Yeah. Yeah. So that's something you need to be careful about. If the batch size is different, it could be, I mean, it's another parameter is going to influence, it could improve your result to have a larger batch size. And then if you just have the baseline with batch size of 1, you could expect that just having a larger batch size would allow you to have better performance. But in that case, they all have the same batch size. Yeah.
Okay, great. Thanks.
You're welcome.
Any other questions? Okay. If not, then let's take this opportunity to thank Florian with some virtual applause. Thanks again, Florian.
Thank you, Florian. That was fantastic.
Thank you.
So we'll be uploading the talk on our YouTube channel later today. So if anyone has more questions, feel free to add them under the video or put it in our Google form. We will follow it up with Florian as well. And I think that's it for today. So we can, let's see.
Thanks, Nadine and C for organizing it. That's great. Thank you.
Thank you.
Thank
you. Thank you, everyone.